Thank you very much for the invitation. Thanks all for sticking around for the last talk, despite this wonderful scenery and weather. So we're looking forward to that. I'm Heiko Runz from Biogen, and I'm, as Ali has mentioned, in the business of drug development. And this business is a really risky one. As you all are aware of, there's about 90% of all the drug programs that are ever being tested in humans failing to meet the market or reach approval. Most of this is because we are just doing not such a good job as we probably should. They're failing in late-stage clinical trials, typically due to efficacy. That means the drug doesn't do what we expect it to do, or to some safety findings that was not predicted at earlier stages of development. There's some recent and emerging evidence that human genetics may help increase the probability of success. There's predictions that drug targets that have a solid foundation in linked to human disease increase the probability about twofold. If you look through all stages of those about 40 new molecular entities that the FDA has approved just last year, about 60% are supported by human genetics. If you look at this data a little bit more closely, you see that the strongest increase in probability of success is for targets where we have a solid link to a Mendelian disease. Most of the time, these are targets where we know fairly well the disease that they are linked to and the molecular mechanisms, how they are actually causing disease. A good example for this, marketed by Biogen, is nusinersen or Spinraza, which is a treatment for spinal muscular atrophy, a devastating neuromuscular disease. Here, we know about research since about 18 and more years that the large majority of cases are caused through homozygous deletion of the SMN1 gene. Humans have a pseudogene, SMN2, that is present in one to multiple different copies, and that leads to a SMN protein that has reduced function due to splicing deficiency because one exon 7 is cleaved out. The hypothesis was done that if we are reducing this abnormal splicing, we might be able to increase the full-length SMN protein, and the hypothesis was that this might really work out to benefit SMA patients in the clinics. And indeed, this is where a lot of academic research has gone in. Ionis Pharmaceuticals has brought forward in ASO therapeutics that Biogen is now has tested in now multiple different clinical trials. This antisense oligonucleotide has been tested in infants with spinal muscular atrophy in multiple studies, has shown to increase motor function and event-free survival. In a pre-symptomatic study, all participants that had received this treatment reached their motor milestones, even preventing the development of disease. There's at the moment a large number of SMA patients on treatment, and this success of this drug has even led to the implementation of newborn screening. A long process where multiple parties have contributed that has indeed now led to a successful therapy for this terrible disease. It's much more difficult often, and despite an enormous effort that went into such a drug development program, when we have to start these days for new targets for complex disease, where we typically know much, much less when we get started with the drug development program. If we tap into the data that comes out of genome-wide association studies, there's a few loci where we can directly relate to the gene, because this is a Manhattan plot. The signals are fairly strong and point just to one single distinct gene that we can believe is thoroughly associated with the disease. Most of the time, these GWAS loci look much, much more complex, and we often just don't know the identity. What is the actual causal gene that links to disease? So that's the challenge that we are typically faced with when we want to initiate a drug discovery, drug development for the more common diseases. What is the actual causal gene in the GWAS locus? And then the question, is it a suitable drug target at all? Just to exemplify the challenges that we have, a variant that you detect in a distinct gene does not necessarily mean this is the gene that leads to the disease. It can also be in a linkage disequilibrium with a gene that, with a variant that is just nearby, and that may impact the expression of that gene as a cis QTL. It may also impact the expression of other genes nearby, or it may act as a trans QTL. That means impact the expression of genes that are fairly far apart. And we, in fact, do not yet know from just this association which is the actual mechanism and what is the gene that confers the link to the disease. We are compiling information and using this information from multiple different omics resources to help make such prioritization. One resource that Biogen had helped to lift off is Open Targets, where a lot of such information is compiled. We're applying this for prioritizing targets from for diseases that we are particularly interested in here, for example, Parkinson's disease, where we can use machine learning and these different omics data sets to make predictions of what are the most likely genes in GWAS loci. However, for the most part, we still don't have the data that really allows us to unambiguously decide what is the causal gene and what is the directionality to use this as a potential therapeutic for the disease that we want to go after. So what is particularly absent or has been absent is information on proteins that are the targets for most drugs, culprits for most of the diseases, and where we heavily rely on as intermediate biomarkers. We hope to find evidence where we can directly make a link for genetics to the changes in protein levels, exactly what we would want to achieve with a therapeutic, and link this then directly to the disease to support our therapeutic hypothesis. We have, in a study that got published last year, teamed up with John Danesh and Adam Butterworth to make a very large-scale profiling of the plasma proteome, where we profile close to 4,000 proteins in about 3,000 healthy blood donors. We established a lot of links between the genome and various protein levels, and don't want to go into any detail of that study, but point out that for a fairly robust number of loci, we could now fairly unambiguously link genetic variants observed in GWAS genome-wide association study to changes in plasma protein levels. And I don't want to go again into this, but just want to show you that such type of information is now really being immediately made use of for drug development. And some of you may be familiar with myostatin, which is a plasma protein that is very tightly linked with muscle development and disease. There's animal models and also humans who are knockout for GDF8, which is the gene that encodes for myostatin that causes muscle hypertrophy. There's no clear link through genome-wide association studies for this. So the question was still, is this a target that can actually be used for general muscle wasting disease? There are associations of with hand grip strength of the myostatin receptor, activin receptor 2B, and that supports the therapeutic hypothesis that if we reduce myostatin levels in plasma, this may indeed increase muscle growth. Indeed, Biogen is following such a program. We have a molecule in a phase 1 clinical study that simulates the activin R2B receptor as a soluble molecule that is modified so it doesn't bind ligands that are believed to cause safety issues. And the hope is that by administering this drug, we will reduce those levels of myostatin, which comes up together with a receptor GDF8, together with another protein GDF11, that we reduce the binding of these two proteins to the receptor, and with this reduce the downstream signal. So with our pQTL study that I had mentioned, we found that interestingly GDF8 and also GDF11 are tightly under the control of a distinct genetic variant that sits in trans, so it means relatively far away from those two proteins, and also interestingly controls another protein in cis, which is called GAS1, controlled by this difficult to pronounce gene here. So by doing conditional analysis, that means normalizing one signal to the other, we could find that this interaction between these proteins does not seem to happen on the genetic level, but actually on the protein level. That means if we a SNP is reducing Gas1, which then on the protein level reduces myostatin. And essentially, this has led to the discovery of an interesting alternative target that may impact the myostatin pathway. We did in silico predictions and mass spec analysis that indicate that these three proteins travel as a complex, and one could hypothesize that the abundance of this Gas1 protein acts in a very similar manner as the drug target that is currently being tested in clinical trials. And that's a hypothesis that we are following through at the moment. So once we have identified a correct target, like the example that I just gave you, we are very much interested to learn how hard do we actually need to hit this target and determine efficacy and toxicity, which we typically do with drugs. However, typically we learn about toxicity and efficacy signals, therapeutic windows, only after long clinical studies, late-stage clinical studies. And what we really would want to aspire is already before we initiate such a drug development program that we can make predictions about the therapeutic success, ideally even before we start with the program and based on the genetic data. For doing this, we are heavily tapping into human genetic data that is being collected in large-scale biobanks. We are using such typically disease-agnostic population information to collect both genetic data as well as real-world phenotypes, which may have been collected as research studies in JIVAs, electronic health records, medical records, or self-reports with smartphones, and then conduct so-called phenome-wide association studies, where we are testing the association of selected SNPs in drug targets that we are particularly interested in to a large range of phenotypes, and then hope that we will find associations between a genetic manipulation of the target with a phenotype that we are interested in, or maybe alternative phenotypes that may indicate that there's other indications we could develop this drug against. Also possibly associations in the opposite direction that could be surrogates for toxicity signals that might show up as adverse events in clinical trials. So I just wanted to briefly guide you through a study that got published last year by Dorothy Diogo, where we tapped into 4 of such biobanks. We teamed up with 23andMe, the first version of the UK Biobank analyzed through Genomics PLC, a precursor of the FinGen study, FinRisk, and the cohort from Children's Hospital of Philadelphia. All of these data sets were different types of real-world data. In the case of 23andMe, self-reports. In the case of UK Biobank, in the most instances, medical interviews, and for the hospital-based cohorts, EHRs, Finnish EHRs, and EHRs as used here. We just did a proof of concept, selecting 25 SNPs near drug targets that we could fairly unambiguously link through JIVAs to a respective disease, and where we also knew this SNP is indeed affecting the gene that we are considering. We analyzed the real-world data from those biobank cohorts together with 44 JIVAs cohorts. And what was, I think, the most challenging part of this entire study was to bring together the different types of phenotype data that has been collected in these different resources, just making a match between heart attack, myocardial infarction, or gallstones and cholelithiasis. That's really challenging, and in many cases had to be made manually. We are still looking for tools to ease that process. Just a quick run through the results of that study. I don't want to go into any detail, just pick out some 2 neurotargets that we analyzed in as part of this LRRK2 and TMM97. LRRK2 is a very, very well-established target for Parkinson's disease. We found back associations for Parkinson's disease for this target, as well as the newer target, TMM175. With this biobank data, we also could find back associations that were previously known to Crohn's disease and IBD for LRRK2, but not the alternative Parkinson's disease gene, which allowed us, however, to make conclusion that this gene might have additional functions in metabolic disease and could have potential adverse events if going after. So that's the type of information we can draw from biobank information to indicate efficacy for distinct diseases. We can also use this to rule out hypotheses. For example, in this case, LGLS3, a gene that had been tightly linked to a various number of cardiovascular diseases. In our analysis, we also find links or association of this locus to Parkinson's disease. And the question was, is there really an association between the protein levels of that galactine-3 encoded by the gene in Parkinson's disease? And by conditional analysis, together with protein QTL data, we could rule out and indicate that this Parkinson's signal is very likely not explained through galactine-3 circulating levels. So these are the analysis that we are doing. We concluded that really these type of analysis fevers across these real-world data sets can inform drug discovery on efficacy, alternative indications, and safety risks. But it's not plug and play. There's not one resource where we could go into and have all our answers that we are seeking for. So Biogen has joined and teamed up with other pharma companies to support setting up the infrastructure that we need for these type of analysis. We are part of a consortium that is sequencing the 500,000 participants of UK Biobank. You may have heard that the first 50,000 that have been generated through Regeneron and GSK have been released just earlier this week. We hope this is a very tremendous resource that the academic research community will make really best use of. We hope that the remainder of UK Biobank exomes will become available over the next few years. FinGen is an alternative cohort that we are helping to build. We have heard about this earlier. We have teamed up with a number of other pharma companies, 9 biobanks in Finland, all coordinated by the Finnish Innovation Fund, where we are generated genotyping data in up to 500,000 Finns, so 10% of the Finnish population. And we are already using such type of data very actively. We are always being asked, is such type of investment, does it make sense for pharma companies? We feel yes, clearly, both on the early side but also on the development side for nominating novel targets. This is resources that we are tapping into daily, essentially, to assess how strong can we indeed validate drug targets based on human genetics. We've conducted here, for example, a large-scale genome-wide association study making use of the UK Biobank data, where we use proxy Alzheimer cases to identify 16 new Alzheimer loci. And even this is possible even with fairly small numbers of actual Alzheimer cases in the UK Biobank. What was particularly interesting for us was that for the first time, APP showed up with these analysis as associated on a population level, previously only been associated on a family-based studies. These type of analysis are also done by academic teams, of course. So if I talk to this with my pharma colleagues, they tell me, well, maybe we want to see more out of such type of investments. And what we are clearly looking for and hope so that these resources can be used for is that we will be changing the way how drugs will be developed in the future. So what we really aspire to do with these type of resources is so-called virtual clinical trials. This doesn't come out very nicely here, but what we hope to do is, well, imagine we would never have to run a clinical trial to test if modulation of the actin receptor 2 or GAS1, the genes that I talked about earlier, are improving SMA. But we could go into these resources and see that in patients with spinal muscular atrophy who are carrying these risk variants might do worse or better than those who don't. And that's the type of analysis we are currently doing. What we also hope, of course, is that these type of resources will spur drug development, but just guiding us to where do we find actually the patients that should be enrolled in our clinical trials, so that we believe might benefit best from a drug. And we are very actively working on testing these type of resources for drug development, and I'll hopefully will be able in short to tell you more about the activities that we're currently doing on the clinical side. And with this, I'd like to acknowledge everyone who has been involved. And again, thank you very much for your attention.